Hello everyone, and welcome to today's lecture. We'll be covering bacterial biofilm formation, discussing what biofilms are, how they form, and how we can observe them during laboratory experiments. Here's what we'll cover in this chapter. First, we'll look into what bacterial biofilms are and the stages of their formation. Then, we'll move on to the experimental method used to study biofilms and analyze the results. A biofilm is a group of microorganisms that stick to each other and to the surface. They are often surrounded by a protective layer called EPS. EPS is a mixture of DNA, proteins, and other compounds that bacteria secrete. This matrix protects the bacteria and helps them survive under harsh conditions. So, why do bacteria form biofilms? Within a biofilm, bacteria can share nutrients and communicate more efficiently. Biofilms offer protection from drying, antibiotics, and the immune system, making them harder to eliminate than individual free-floating bacteria. Let's talk about how biofilms form step by step. In the beginning, bacteria are mobile. They move around using flagella. They swim to find a suitable surface rich in nutrients. Once they find that surface, they attach to it and undergo a big change. They stop moving and start producing a sticky substance called the extracellular matrix. They also stop separating after division, forming chains of cells. As more bacteria join, the group grows and forms thick layer. This is a developing biofilm. The matrix holds the cells together and protects them from the outside environment. When the biofilm matures, some of the bacteria form spores, which help them survive under harsh conditions. Eventually, certain cells begin producing small molecules like D-amino acids and polyamines. These molecules break down the matrix, allowing some bacteria to escape and move elsewhere. Then the cycle begins again. Model bacteria find a new place to settle, and a new biofilm starts to form. Now let's go over the experiment used to study biofilm formation. First, take a 24-well plate. Add 1 milliliter of TSB medium to each well. For blank wells, add 1.5 milliliter of TSB. Inoculate each well with 500 microliter of bacterial culture. Incubate the plate at 37 degrees Celsius for 48 hours to allow the biofilm form. After incubation, we quantify the biofilm using crystal violet staining. First, measure the OD600 of planktonic, which is free-floating cells. Remove the supernatant and dry the wells for 10 minutes. Rinse with distilled water and dry again for 10 minutes. Add 50 microliter of crystal violet to each well and stain for 10 minutes. Wash again with distilled water. Dry for another 10 minutes, then add 1.5 milliliter of 95% ethanol. Finally, measure the OD600 again to estimate the amount of biofilm formed. As a result, we can calculate the BFI, biofilm formation index. So the formula is here. The table shows how we categorize the results based on the OD values. To summarize, biofilms are protective communities formed by bacteria. They help bacteria survive harsh conditions and resist treatment. We learned how biofilms form and how we can measure them today. Thank you for your attention.